All protocols duly observed and respected. My name is Bahauddin Salisu, representing my research team to present you our research work with the title A Polyphasic Chemotypic and Molecular Characterization of Aspergillus section Flavi Contaminating Food Grains and Poultry Feeds in Malaysia and Nigeria, with a paper ID BIOSPEC PA61. Now, brief overview of the study. Aspergillus section Flavi ASF contaminate about 20% of the global food, resulting in 4.5 billion people being exposed to aflatoxin, which contribute to about 20% of annual global cancer deaths, with most of the cases occurring in Africa. Now, according to IARC, regional aflatoxin Aspergillus section Flavi data is needed because occurrence, toxicity, genotype of ASF are not fully elucidated in most regions. New highly species are continuously discovered, and as a result, data of local ASFs is needed for effective aflatoxin control. Now, the conventional screening method are affected due to environmental factors which affect phenotypic appearance of isolate, leading to inaccurate identification. On the other hand, the molecular method is also limited due to genome similarity, which lead to inaccurate identification using this method alone, because, for example, the genome of Aspergillus flavus and Aspergillus plasticus are about 97 to 99% similar. Therefore, it is suggested that a combination of method, multivariate technique comprising phenotypic, chemotypic, and molecular characteristics should be used for accurate identification of species, and this is needed to improve regulatory strategies of aflatoxins. Now the study objectives and rationales. Now, in our previous study, we isolated about 74 ASF from 666 samples of poultry feed and food grains in Malaysia and Nigeria. The question that arises is that what is the toxigenic potencies of this isolate? And this is what led to this research. With the question, what are the toxigenic potencies, chemotype, genotype, and actual species names and phylogeny of the isolated ASF? To answer this question, we set the following objectives in this study: to determine the strain types and aflatoxicity of the isolate, to determine the chemotypes, species names of the isolate, and to confirm the species name and aflatoxicity of the isolated ASF using molecular methods. Now, to achieve this, we set the following experimental stages, which I will explain in the during the course of my discussion. Now, the first stage of our work is phenotypic characterization, in which we subculture all the 74 ASF in 5 different culture media, and we observe for their type of celerosea, based on which we are able to divide them into 3. Those that produce celerosea, NS, those producing small celerosea, and those producing large celerosea, as you can see in this picture. The second stage is aflatoxin screening, in which we observe all the plate under UV light to detect aflatoxin. As you can see, the first 2 plates do not have aflatoxin fluorescence, while the second one have aflatoxin fluorescence, which and therefore they are regarded as those for aflatoxigenic species. This is the summary of the phenotypic characterization. As you can see, PDA media produce the highest number of aflatoxigenic isolate, followed by SDA media. Now, in summary, 41 isolate were aflatoxigenic, while 33 isolate were confirmed non-aflatoxigenic, and this result correspond to several other literature that report aflatoxigenicity of Aspergillus section Flavi in Nigeria and Malaysia. To confirm the aflatoxigenicity of the isolate, we optimize a high-performance liquid chromatography that can be able to quantify 5 different exometabolites that are used in characterization of Aspergillus section Flavi, with cyclophosphoic acid and 4 aflatoxins. Now, this is the summary of the quantification. We have the exometabolite, and we have the number positive, and we have the concentration. And then based on this, we were able to categorize the isolate into 6 different chemotypes, with chemotypes 3 and 4 being non-aflatoxigenic. And then based on this chemotypic grouping, we now identify the isolates using references from literature, in which 60 isolate were confirmed as A. flavus, while 14 isolate were confirmed as Aspergillus oryzae. For the objective 3, which is the molecular identification of the isolate, we carry out two sets of monoplex PCR using two most used primary identification genes, that is the ITS gene and beta tubulin genes. In this figure, A part of the figure shows the amplification of the ITS primers, while the B part shows the amplification of the beta tubulin primer. Now, just similar to the chemotypic identification, 60 of the isolate were confirmed as Aspergillus flavus, while the remaining 14 were Aspergillus oryzae, and all the sequences were deposited in the NCBI, where they were given an accession number. Now, finally, our result correspond to several other studies that report similar findings in literature. After confirmation of the species names, we carry out multiplex PCR using 4 important aflatoxin biosynthesis gene in order to confirm the aflatoxenicity of the isolate. Now, in this figure, as you can see, this is the first set of the multiplex containing 2 primers, apple P and apple D primer. So this is the pattern of the amplification of the aflatoxin of the 2 aflatoxin genes by the isolate against the positive control and negative control. Now, in the second figure here, we have the second set of the multiplex PCR with the 2 primers, apple R and apple M primer. So you can see this is the pattern of the amplification of the primers by the isolates. Now, all in all, a high number of the large number of the species were confirmed aflatoxinous, and the result correspond to that of the chemotypic identification. And then this result also correspond to several other results in the literature, which shows the amplification pattern of Aspergillus section Flavi. To conclude, this study characterized 74 Aspergillus section Flavi using polyphasic approach. All the 6 chemotypes of ASF were present among the isolate, with the aflatoxigenic group being significantly higher than the non-aflatoxigenic isolate. The aflatoxigenicity of the isolate does not correlate with the Celeratium morphotype, and both toxigenic and atoxigenic isolate were phylogenetically clustered in the same clade. Data obtained in this study claim attention for probable co-exposure to fungi and mycotoxin to the people in the sampling area. Acknowledgement, we are thankful to the Ministry of Higher Education Malaysia for sponsoring this study. These are our references, and thank you.